Um, it's my great pleasure to introduce today's speaker, Dr. Di Yu. So Dr. Yu is currently a professor of immunology in the University of Queensland in Australia. So Dr. Yu got his training with Carola Vanusa in Australian National University. In 2011, he started his own lab, first in Monash University, and then moved to the University of Queensland. Dr. Yu has a long history in studying T-cell biology and its translational application, especially on follicular helper T-cells. So as we all know, the follicular helper T-cells play a very important role in B-cell development and antibody production. So Dr. Yu has published many influential papers and was listed among the highest cited researchers in the field. I think one of the most exciting works is his recent discovery on leptin, the famous hormone in obesity, can promote follicular helper T cell function and support vaccine response through the production of IL-21. I think Dr. Yu will cover some of the story in his talk. So without further ado, let's welcome Dr. Yu.
Okay, thank you very much, Xiaolei, for a very nice introduction, and in particular for Xiaolei and Wu Chuan, who have organized this fantastic seminar series, and we're able to share and communicate with most of the Chinese immunologist community around the world. So today, I will share some of our research on the topic of follicular T cells, which is the topic we have studied over almost 20 years now. Before I get started, I would like to recap how we come to the topic of T cell help is important for a B cell function or humoral immunity. Many of you would remember in 2019, the Albert Liska Award were given to Max Cooper in the United States and Jack Miller in Australia. And both of them made a significant contribution for the immune system and immunology for their discovery how the B cell, T cell development and their fundamental function in the immune system. And Max Cooper for his discovery of the B cell and B cell development, and Jack Miller, many of you would consider he was contribution would be the T cell development. Nevertheless, Jack Miller not only discovered the thymus is important for T cell function, T cell development, but also demonstrate thymus is also required for generate efficient antibody response. So this was the citation of his publication in the later 1960s. And the thymus might influence antibody production in mice. Only mice that receive cells from the thymus, which are T cells, and bone marrow, which are B cells, can produce proper antibodies. And the study also reviewed the bone marrow derived cells, which are B cells, produce antibody, but only with the help from the thymus derived cells, which are T cells. So the concept of T cell help for B cell or antibody response is not known as being discovered almost 50 years ago. But there's quite a few major questions about the T cell help for B cells, such as what T cell help B cells? Is there a phenotypical or functional subset for T cell or in T cell to help B cells? If so, what's molecular mechanism or molecular programming to determine behavior function of T cells? And the question is also in terms of function, how T cell help B cell in antibody response, such as that through the cellular interaction or this through secrete soluble factor for B cell. And eventually, by understanding such knowledge, we try to understand, try to infer this knowledge to the applications such as how the T cell helps involved in the immune-mediated disease, and probably more important at the moment is how this can make a better vaccination. I want to give you a very simplified model how the T cell help B cells, largely what we have known to date. So this is the immunofluorescence section staining of the tissue from a mouse lymph node. In the peripheral, you can see this green staining. They are all, sorry. The peripheral staining is the IgD. This is a naive B cell marker. So particularly, you can see this is we call the primary B cell follicle. And then we have G07 staining red here. They are staining germinal center. And the follicle contain the germinal center we call the secondary B cell follicles. In the middle of the lymph node, we didn't staining, but these are flow of T cells. They call T cell zone. So the T cell zone is surrounded by the B cell follicles. So during an immune response, let's zoom in. The T cells get activated, and they can migrate from the T cell zone into the B cell follicles primarily based on upregulation of the chemical receptor called 6R5. This was a seminal discovery by Jason Sister's group in 1999, about 20 years ago. So in the TB border or in the B cell follicle, 6R5 positive T cells provide helpful B cells, which are also primed by the antigen. So they can have a base level antigen recognition by the B cell receptor. So with this help at the co-stimulation or cytokine, the antigen priming B cells can expand and form the germinal center, here staining the red. So within the germinal center, TFA cells provide not only just supporting signal, but also selection signal. So only very small fraction of the germinal center B cells can be successful and positively selected to differentiate into memory B cells or antibody secreting cells. So this is what a very simplified model how the T cell upregulate 6R5 support the germinal center and selecting germinal center become antibody secreting cell and memory B cells. So this is a very nice model. How we came to this model, a little history. So I mentioned in the 1960s, it was already known that T cell helper is important. And later on, the germinal center was discovered by the histology staining, and now these are proliferation of B cells. And from 1980s, from early 1980s, a study has demonstrated the T cells within the germinal center. Now we know follicular helper T cells have a different phenotypical marker expression. But less is about this because the technological limitation. As I mentioned, the 6R5 discovery by Jason Sister is really setting a cornerstone for the TFH study. And you can see this early study is look at immunization. So on one axis, activation marker CD44, and then staining 6R5, which is the chemical receptor for XL13. So a large number of activated T cells are upper 6R5, which can migrate towards the germinal center, have a higher staining of 6L13. And this demonstrated by the chemotaxis action, demonstrated 6R5 positive, not negative cells can migrate towards 6XL13. So soon after, use 6R5 as a marker, a few groups able to isolate human 6R5 positive, negative, and activated CD4 T cells, core culture of the B cells, and demonstrated 6R5 positive cells were far more potent than 6R5 negative T cells, enhanced B cell for antibody production. And in the 2000, that's a year, this follicular helper T cell, the concept was coined. So if many of you probably remember in early 2000, before we have an RNA sequencing, the microarray was used to do the gene expression profiling. And this is just one of the studies demonstrates 6R5 positive T cells have a very distinct gene expression profiles compared to other T cell subset, such as they can express 6XL13, 6L5, and also you can see have ICOR, CTL4, and PD1 very high in the TFH cells, and also you can see BCL6 is highest. And that is largely from the mouse study human T cell study by co-culture or gene expression. And I was getting involved in this study because my group, when I was working with Carola Vinusa, we parallel discover the mouse TFH cells. And this is from the mutant strain. I'm not going to the details, this has been a long time ago. But what we have seen here, this mice develop spontaneous germinal centers with lots of CD4 T cells infiltration, without antibody production, and enlarge the lymph node and spleen. And you can, well, you probably cannot see here, but the general, the mouse T cells show a comparable TFH signature as reported human TFH cells. So that is the characterized for both mouse and human TFH cells. And later on, this initial study demonstrated IL-21 is one of the major effector molecular or cytokine produced by TFH cells, which support the germinal center without IL-21. Of course, dependent on experiment, and some experiments showing there's some stronger defects on TFH, some actually is less defects. But compared to other cytokine, IL-21 is considered one of the major TFH signature cytokine. I think one of the critical year for all the people working on TFH is 2009, and the discover BCL6 as a master transcription regulator for TFH really set the scene for the whole field and led to the boom of the TFH study. And I think one of the demonstration of this is before 2009, there's quite a few T cell lineage and Treg or other Th1, Th17, such as this review written by Simon Sakaguchi. TFH wasn't in the picture, but in the 2010, after the BCL6 was discovered, the community largely acknowledged the importance of TFH and the BCL6 guiding the differentiation as a separate lineage within the CD4 T cells. And we actually celebrated this, and we was lucky. I think that was before the COVID-19. We was able to organize a meeting in China with a lot of people doing the TFH study and to celebrate 10 years of the BCL6 study, and of course discover future and the field how we move towards. How the BCL6, why the BCL6 is very important. I think there's, of course, without BCL6, there's no generation of TFH. But the BCL6 also reveal a very interesting multiple mechanism of action for TFH cells. This is the review article by Shankrati. There's a few, but I want to just point out, so first of all, BCL6 is largely the transcriptional suppressor. So BCL6 can suppress the alternative differentiation of CD4 T cells into Th1, Th2, and Th17. And on the other hand, the BCL6 is also suppressed some other transcript regulators, such as you can see BLIMP1 and also ID2 and RAMS. And because ID2 and BLIMP1 is a strong suppressor of 6L5. So through this, the suppressor of the suppressor, BCL6 able to operate 6L5. But many of you, particularly for those working on CD8 cells, understand the effector, the PRDM1 encoding BLIMP1 or ID2 are the strong transcript regulator for the CD8 effective differentiation. So this actually very similar between the CD4 and the CD8. So the CD8 cells also have a BCL6 and get involved in the regulate the fatal decision. On one hand, the BCL6 together TCF1 operate 6L5. On the other hand, they can turn the CD8 cells into more memory phenotype and downregulate the exhaustion in this population. So I think this BCL6 and the related transcript regulator form a regulatory circuit in both the CD4 and the CD8. And particularly for the CD8, there's a lot of interest in here and a few publications within my group or from Yeli Lin's group, Rafi Amin's group, and this particular study by Wu Tuoqi, so at the same year, we support this idea that 6L5 positive and the TIM3 negative and PD-1 positive cells have more important role in memory generation, and in particular, some of these studies demonstrated that responding population for PD-1 therapy. So in summary, there's a large number of studies. There's a major contribution from a few groups, including Carola, Sankroti, and Chendo and Haiqi, and many others. Also, some study from our group and demonstrate a few answers for the question: follicular T cell are functional subset with a distinct phenotype, and they control the B cell health, and they rely on BCL6 for their generation, and they need cell-cell interaction, but also secretly soluble factors such as IL-21. So today I will mostly talking about the role of TFH help in immune-related disease, which is autoimmune disease and vaccination. The first part I will give a very brief, which has been published a few years ago, the role of TFH help in immune disease and vaccination. First part is talking about pathogenic TFH cells, which in autoimmune disease. Before I move on, I would like to mention, because if we are all working on the mouse model, it's very convenient. We can take spleen and the lymph node. We can look at the lymph node architecture and profile. But for the human study, it's challenging because we have to move on from the lymphoid organ into the blood to understand TFH signature. And from this particular study, we demonstrate there is a TFH signature in the blood, and they also express 6L5, may not as high as those in the human tonsil, but it is particularly this PD-1 high, 6L7 low population. You can see this is a mouse model post immunization. Then this is a transient increase of this population indicate TFH activation. And we can see this is from human post-vaccination on day 7. And also in the lupus patient, they can continue activation of TFH signature by this PD-1 high, 6L7 low, and 6L5 positive population. Some of you may consider why lots of people studying vaccine and T cell response always look at day 7. That means because some of early study, including ours, demonstrate day 7 for best the TFH signature in the blood. So use this tool, many of group, including ours, such as this one, demonstrate the TFH hyperactivation in autoimmune disease. So a lot of types of autoimmune disease, they can have a manifestation in many different organs. And because the excessive TFH, we have discussed in the mouse model, but they can largely support autoantibody production. Instead of a protective antibody, such as infection or vaccination, the autoantibody can recognize self-antigen, mitigate the tissue damage and the inflammation. So the TFH have a strong role in autoimmune disease. And how we can suppress TFH activation. This is a diagram in my review and demonstrate we definitely can suppressed by the co-stimulation signals such as the CTLA4, which has been FDA approved to treat rheumatoid arthritis and also testing type 1 diabetes. And also can suppress the IL-6 signal because this signal was required to positively induce TFH. On the other hand, this the study in the mouse model revealed the IL-2 is a very strong signal, can suppress the TFH generation because IL-2 through STAT5 can suppress TFH generation. So based upon this mechanistic study in the mouse model, sorry. What's going on? So based on the study on the mouse model, we collaborate with rheumatologists in China to initiate the first clinical study using low-dose L2 therapy in the lupus patient. And you can see this is the 3 treatment cycle using low-dose L2 for the lupus patient, each cycle for 2 weeks, and then we have a blank for 2-week result at the time was in case of side effects. So you can see they have a strong climbing of the response rate until 90% for L2 therapy, and this is just a 1 cycle to quickly resolve, at least for this skin manifestation. And we can see the clear reduction of TFH cells by low-dose L2 therapy, and because L2 also suppresses Th17 cells, we can see also resolve the Th17 hyperactivation to some extent. Of course, one of the major mechanisms of action is also to expand the Treg cells to introduce immune homeostasis and tolerance. So this was the open-armed clinical trial, and that was followed up by a double-blind clinical trial to demonstrate low-dose L2 therapy was effective and safe for lupus. Now it's recommended, at least in Chinese Society of Rheumatology for patients, and many of patients, even some patients I know, were able to benefit from this therapy by low-dose L2. Now I want to move to major part of my talk is how we are consider the TFH and protective antibody response. There's a few questions how the TFH have a role in protective antibody response. The first of all, why individuals have a different vaccine response? Some are good, some are poor responder. For those who don't have sufficient response, how we can lift the magnitude for vaccine response? Of course, we are also consider how we can prolong the immunological memory so the protection can sustain. And also, we are also consider the mechanism what regulates antibody selection so we can generate the more broad neutralizing antibody. This is important for COVID-19, but of course, it's a key topic for develop HIV vaccine. If you look at the antibody response by vaccination, we have this kind of 2 wave after vaccination. And the first few days is more like a natural IgM antibody production, following by the general center mediated and TFH supported higher affinity antibody production from probably 1 week. And then that's led to the protection and the infection. We have a much quicker antibody production, and that can mediate a protection. But that is ideal situation. In some individuals, when the response was not so good, they probably don't sustain the antibody response, or the antibody response was in the very smaller scale, and the protection will be compromised. So the first part of this question, we try to understand the individual difference is, we try to understand what's determined the TFH. I'll come back to this. And this was a study I strongly collaborated with Li Wei-Lu from Hong Kong University, and it was conducted by 2 postdocs, Michael and Anthony, in my group. And Michael was a PhD student in Li Wei-Lu's group. I just want to show one piece of data which already also published by other groups. So this is influenza vaccine. And you just quick look at this. This is a 3-strain vaccine. Now we also have a 4-strain vaccine. So this is the level of TFH activation on one axis. On the other axis is the antibody titer by each of the strain. And you can appreciate there's a strong positive correlation between the TFH activation. And many studies indeed demonstrate the activation of TFH, even though antibody is produced by B cells, but the bottleneck or the limiting factor is the activation of TFH cells. And then the question is, what determines the TFH activity in the general population? We are not talking about a patient with immunodeficiency, or we're not talking about with a compromised immune system, such as the immunosuppressive treatment. We are talking about the healthy individuals. And even in healthy individuals, the vaccine response are not always perfect. And particular highlight by our understanding of aging study is when we are young, we have much better antibody and vaccine response. And for the case of influenza vaccine, the protection will be 80–90%. When we are getting older, the protection can quickly drop to below to 50%. So they do have a non-genetic determining factor to regulate antibody response. One of them, I think was started from a Liver-Luz group, was the leptin signal. So leptin was a hormone largely produced by the adipose tissue, and a major role previously was studied in the metabolic system because leptin can target brain and regulate the food intake and also antigen expenditure, basically the function, the fundamental function of the metabolic system. However, leptin also have a receptor ubiquitously expressed by the immune system, and some early studies, this is a review published recently, give a sense, is the leptin has this leptin receptor expressed by lots of cells, including T cells, both CD4 and CD8 T cells. They have this pathway. One is the STAT3 pathway mainly, the other one is mTOR pathway. And many of you know these are the two pathways important for T cell activation and differentiation. And on one hand, the leptin has been shown to also require for Th1 and Th17 fully functional differentiation. Some studies demonstrate that leptin actually can enhance the Treg cells. So the lower leptin has been associated, of course, obesity, but on the other hand, insufficient T cell function. But higher leptin have some indication for the autoimmune disease, but this needs to be examined more thoroughly. So what we have started to look into is we put the leptin in the in vitro culture, the T cell, which is the human T cell to start with. What we have detected, leptin strongly induced IL-21 production. Probably IL-21 is strongly induced by the STAT3 signal. And also, co-culture with the leptin, we can see they also can increase 6L5 expression. I appreciate it's not very strong, but this is significant. In vitro generate 6L5, particularly human cells, is could be challenging. But this towards a positive regulator as a leptin for TFH signature. So before moving on the mouse study, was asking why there's a true association between the leptin and vaccine response. So what we have seen here, this is one of the first group who examined. So this is a middle age. That's why they only have a few poor responder. We can see a spectrum of leptin, which is really cool because the leptin have a really wide spectrum to cover over almost 100-fold difference. But we can detect non-responder have a generally, I think it's not very strong, but a generally lower leptin level. But if we divide the whole population into this, we call the low quartile because the leptin can vary from age. So there's no standard normal leptin level, but we can, for the individual age range, you can go this lower quartile. We call it low leptin, and then this called the sufficient leptin. And in the low leptin, we got 20% rate of non-responsiveness. But when the leptin level is high, almost all the individuals responded to vaccination, this influenza vaccine. Of course, it's only a few non-responders. It's very hard to convince ourselves, so we move on to the older population. So this is the older population. Now we can see a more strong phenotype is again, the leptin level is lower in the non-responder, but now for more than 50%, the low leptin quartile not responded to the vaccination, and then on the leptin sufficient population, this dropped to like 60%. So it's a very large difference when the leptin level. And we also looking at some the medical students who received HPV vaccine boosting, even though they have a previous, but they don't have a sufficient level, probably also due to their poor TFH or poor HPV response. But even in this population, we also see the leptin associate with the lower vaccine response. Again, in this population, the lower leptin largely non-responder, but with a higher leptin, the response rate is much better. So taken together, this demonstrates there seems a link between the beta leptin, not that the beta leptin, the higher vaccine is say the lower leptin actually have a poor vaccine response. So we want to test whether this is a peculiar association or this a causation for the TFH activation in the healthy individuals. So we first look at this in vivo, use the mouse model. We knock out the leptin receptor in the T cell, use the CD4 CRE, and expose mouse for the influenza not vaccine, but just infection to look at TFH. So Treg cells and the general CD4 activation looks largely normal when the leptin signal is lack of in the T cells. However, there was significant reduction of TFH and particularly their IL-21 production. And this is accompanied by the B cell defects such as GL7 staining of germinal center, you have twofold reduction of germinal center, and also reduction of antibody sequence cell staining here. So demonstrate the T cell intrinsic leptin signal limit the TFH differentiation and the function, and led to the antibody response was compromised. So in the human being, we hardly have the total lack of the leptin signal. So to make it more physiological, we test in the fasting model. So fasting can reduce the leptin level. So we alternative day fasted mice with the so the leptin level was reduced. And we particularly choose the day 5, day 10 as the period of fasting because this is the key time, the antibody response after vaccination. So we give it 2 vaccination on day 0 at day 10, but then this period of time is do the fasting. We have another group is we call the leptin injection. So basically we have 3 groups. One is the fasting, and the other 2 groups, one is the normal diet, and the other 2 are fasting, one with PBS as control, the other one give back the leptin to see whether the leptin is the key difference, because the fasting definitely can reduce many other metabolic function of the system. So from TFH perspective, with staining 6alpha BCL6, TFH reduced by fasting and can recovered by leptin supplementation, which is the same for germinal center B cells and antibody production measured by the antibody sequence cells. And for this experiment, we particularly want to test the vaccine-mediated protection. And we see after 3 weeks, we allow the mice to all back to the normal feeding, and they recover the weight loss or any other thing. And now they challenge it with the influenza vaccine. And what we can see here, or challenging with the influenza infection, and what we can see here, compared to the normal feeding, the fasting itself largely reduced the vaccine-mediated protection, but give the leptin back can recover the leptin-mediated protection and the vaccine-mediated protection. So I just want to brief summary here. If you have more interest in this mechanistic study, such as signaling, you can go back to this paper. We demonstrate leptin through leptin receptor going to sensory and mTOR pathway to regulate the TFH signature, and that is associated in not only the mouse model for infection and the vaccine-mediated protection, but also in the human population. And I really like to emphasize this study is because leptin produced by the fat tissue, but act as indicator for the energy and nutrition reserve. So those individuals, when they have a lower leptin, they actually have probably limited the energy and nutrition. And it becomes very luxurious for them to have a full magnitude activation of immune response. As many of you know, both the innate and adaptive immune system needs a lot of substance and energy to be fully activated. So we would rather from evolutionary perspective to reserve some of these for the fundamental survival of the human rather than fully activation of the immune system. I believe leptin is one of these indicators to connect the metabolic and immune systems. Now, the very natural next question is how we can make the vaccine better if we do know the TFH and TFH less activation or function is a limiting factor for vaccine response. So a very natural thought would be, okay, let's make a TFH survive better. They can last longer because TFH is also a transient activation. Then this have some function and eventually get reduced and resolve the germinal center response. Can we survive the TFH longer? So make the germinal center more potent and get a better vaccine response. So that is the initial thought. And this was again the collaboration study by Anthony and Ying, who is a trainee clinician and come to my lab as a postdoc from Zhen Liu's group from Tongji Hospital in Wuhan, China. What do we understand of the immune cell death? And apoptosis has been largely studied for the immune system. It was demonstrate important for the T cell, B cell development in thymus and the bone marrow, and also in the peripheral activated T cells undergo apoptosis, and also in the germinal center, the apoptosis regulated germinal cell selection for B cells. So there are programmed cell deaths that don't trigger very strong inflammatory response to maintain immune homeostasis. Of course, we have other ferroptosis and apoptosis to give a more inflammatory response for immune activation. But there are more type of cell death pathways or type of cell death has been discovered. You know, a lot of them, some of I don't understand, and some of them I cannot even pronounce the name properly. But when we look at the TFH, we do a simple experiment. We just use the bone marrow, mixed bone marrow reconstitution into the randomized with wild type or BCL2 transgenic. You know BCL2 antagonized apoptosis, so BCL2 transgenic strongly suppressed apoptosis in immune cells. So when we look at this mixed bone marrow chimera, so the germinal center, the B cell received B cell transgenic very strong expanded. That means, as we published, the germinal center was suppressed, was regulated by apoptosis. But we cannot do this because this germinal center, even there are a large number of germinal center, but the selection was a problem. So we cannot directly target the B cell for enhanced germinal center. But what surprised us, if we lose the BCL2 transgenic, look at TFH, we don't see much difference between the wild type and BCL2 transgenic T cells in terms of TFH number. So TFH seems not particularly regulated by apoptosis. So what is the cell death pathway regulated TFH cells? Weiping presented the ferroptosis very nicely last week on Thursday night. So I give another very brief introduction by this. So ferroptosis is a new form of programmed cell death. This is really dependent on iron. So iron can paradoxize particular lipid and particular lipid in the mitochondria. So the ROS together with the iron and can paradoxize the lipid in the mitochondria without the repellent, the damage accumulate will compromise the function of the mitochondria, and that can lead to the cell death. And one of the major is the glucosamine paradoxidase 4, which is required to reduce this paradoxidation and maintain function and prevent ferroptosis. And as you can see, this is not an immune cell, this is a cancer cells, but you can see with the induction of the ferroptosis, the mitochondria lose the morphology. They become smaller, and they begin to lose the integrity of the mitochondria, the membrane. So when we compare the morphology of non-TFH and TFH cells, what we can see here, the TFH, the mitochondria, they are smaller, they definitely shrink, and also some integrity issue, and this is just quantification of their mitochondria size between non-TFH, TFH cells. And if we lose the more clear system with OT2, so OT2, they generate antigen specific similar TFH and non-TFH cells, and we can appreciate 6L5 positive cells have far more lipid ROS. And this is just a quantification. Show in this immunization, TFH has more lipid ROS and shrink mitochondria. So begin to suspect the ferroptosis was the major regulator for the cell death of TFH cells. And we then move on to test the GPS4 knockout mice. As many you know, the GPS4, just mentioned, is the major regulator to prevent cells from ferroptosis. And without GPS4, those cells susceptible for ferroptosis will become deficient and dying. So we again use CD4-CRE, the GPS4 deficient mice, and we give the immunization and look at 2 time points after immunization. And we can see there's a large number reduction of TFH without GPS4 in the T cells, and particularly in the late time points, almost gone. And even more striking, we look at the germinal center staining. We can see without GPS4 in the T cells, there's no germinal center formation. You can see there's no red staining, and there's no germinal center formation without GPS4 in T cells. And the total antibody was largely reduced. And probably have some basic antibody level, but high affinity antibody most come from the germinal center, totally absent. So let's demonstrate TFH rely on the GPS4 to prevent themselves from ferroptosis. But you may ask why TFH, because we didn't see other CD4 cells are more important. So if you look at what induced the ROS in the T cells, as many other people have demonstrated, the TCR signal, CD3 signaling is basically very potent to induce the ROS in the T cells. So this is in vitro. And in vivo, what we can do, we introduce this antibody targeting system. So we use anti-DAC205, have a receptor highly expressed by germinal center B cells. And we inject this antibody, and they can present more antigen to the TFH cells. You can see an increase with the ROS. And from in vivo, what we have seen, there's a 2 types of immune cells. Not 2 types, also 2 situations of TFH cells. One is TFH, they conjugate with the CD19. That means these are more germinal center B cell conjugate TFH, or less are conjugate. They also have a more ROS. So all this study, I have other data I didn't show today because in interest of time, we demonstrated TCR signaling is the major signaling strongly upregulated the ROS in the TFH, and that made TFH more susceptible for ferroptosis. And that is one of the reasons why TFH compared to TH1, TH17, because they have constitutively TB interaction. That's why they are more susceptible for ferroptosis due to constitutively TCR signaling. Now, with this knowledge, how we can mitigate the ferroptosis and enhance the function. So to prove of concept, we use the ferrostatin. So ferrostatin is an inhibitor for ferroptosis. We give a few doses of ferrostatin during day 5, day 13, and look at the mice at day 14 after immunization. So TFH increased by ferrostatin treatment, enhance the germinal center, and enhance antibody production. That is cool, but not yet to be translated into human being, because this is just purely not a therapeutic compound. It's an inhibitor for experiments. Can do something else. We pay attention to GPS4 because GPS4 is selenium protein and really rely on selenium for their anti-ferroptotic function. So we fed mice with 1 month of selenium, and then we immunize with immunize to antibody response. And with the selenium feeding, we can get enhanced TFH, enhance the germinal center. Not only the number or the amount of antibody increased, but also the affinity maturation was increased. So this is pretty promising. That means we have a very straightforward way can directly test it in the human being. And before testing in human being, we want to look at whether there's a conserved mechanism between the mouse and the human TFH cells. Now we're getting our human TFH cells, and they also have increased the lipid ROS, and also the lipid ROS substrate also increased in the TFH compared to non-TFH cells. Oh, sorry, I think I missed this is CD45 RA, and this is 6L5. And also we use the inhibitor to confirm again the germinal center B cells more susceptible by and rescued by the apoptosis inhibitor, and the TFH survival was more rescued by the ferroptosis inhibitor. So demonstrate the human and mouse have this very conserved mechanism for TFH is regulated by ferroptosis. Now we going to the controlled human study. We give the human being 30 for each group, a month of selenium and also a month after vaccination, just 1 dose of vaccination, influenza vaccination. There was an increase of blood, a serum selenium level, and the GPX4 level also increased compared before the selenium supplementation. So they can increase and kind of plateau after 1 month. And we can see there's an increase of the PD-1 high TFH activation TFH cells as mentioned before. And we have, I would say, significant, but I would say it's a modest increase of antibody response. So this is proof of concept. We can definitely target ferroptosis pathway for better vaccine response. The consequence for TFH is good, but antibody was not drastic. There's at least 2 reasons. One is probably because this is all very young adults. They are only 23 years old in average age. So they may already have a high and cap how much antibody response they can gain. For many of antibody response, it's not more the TFH better, but the TFH can support magnitude, but particular for those has a susceptible, that's more immune compromised or the aging. So we need to move this to test in the aging population. On the other hand, maybe selenium itself as a dietary supplementation maybe not is the best, efficient way, but we are also testing other therapeutic intervention for this strategy. But I think this is very exciting and provide a novel venue to enhance vaccine response. So I would like to summarize the second part of my talk. Is this is the German Center. And particularly, this frequent TB interaction induces strong TCR signaling, which can lead to high ROS in the TFH I didn't show today and associated with the high lipid ROS. And this lead to the susceptibility of TFH for ferroptosis. And GPS4 is critical to protect TFH cell from ferroptosis and use selenium and potentially other intervention strategy. We can protect TFH from ferroptosis, and this can lead to the better antibody production. So that is the summary. TFH from ferroptosis require GPS4. We make it survive better and better vaccine response. So I would end up by saying my group are really interested with CD4 and particularly TFH. We will also have some interest with CDA, but that is a less developed. Hopefully next time we can talk more about this. But really, those are both pathogenic role of TFH and protective role for TFH cells. We have done quite a bit of the TFH in autoimmune disease. I mentioned something, some study today. We also collaborate, look at TFH in the allergic disease. And also interesting, TFH is a major reservoir for HIV, which that's why we discovered these 6R5 positive CD8 cells and control the TFH infection. And also TFH have an interesting role to support the B-cell lymphoma. That is their pathogenic role in TFH. Of course, now we are more concerning for the protective role of TFH. I tell you 2 stories about how the metabolic system, such as leptin or the selenium and the GPS4 axis regulate the vaccine response. We also study how the TFH subset maintain immunological memory and how they control the antibody reservoir, which hopefully I can tell you in future. Before ending, I would really like people make a major contribution. Show some of them during my talk, and Michael, Ying, and Anthony make a major contribution for the 2 studies I mentioned with other people in my group to contribute. And the 2 studies mentioned today is vaccine studies collaborated by Li Weilu's group from Hong Kong University and Zhen Liu's group from Tongji Hospital. And particular, Zhen Liu's group made all the effort to do the selenium supplementation clinical trial in Wuhan. And some other human clinical study was collaborated from Catherine from University of Melbourne and Barbara Ham Institute, Michelle Zell from Cambridge, and also CSL by sample provided by Steve Rockman. And that is my founding body. I would like to conclude my talk and would love to discover a few thoughts with you. Thank you very much.
Okay, this is really a wonderful talk. Thanks so much, Dee. So I see actually Tran has raised his hand.
Hi, Dee. It's
Hi.
great. It's great talk. I mean, I think it's from the basic to translational. It's really comprehensive. So I have two questions. One is related to the leptin story. So you mentioned that the vaccination obviously have correlation with the fooding condition, whether you fast them or you give them lectin. So I think, I mean, I didn't read your paper, but I just want to double check if you ever tried to test the different feeding condition on the leptin receptor knockout mice. Because obviously leptin receptor can express on a lot of cell compartment. So you cannot exclude that these vaccination consequence is indirect by other cell type, but not T cell per se. So that's why I want to check if you, let's say if you fast an animal and then on the leptin receptor knockout and then do vaccination, do you still see the effect?
Right. So we haven't done that experiment. That's definitely a brilliant experiment to do. There's one reason for that is that fasting experiment was done by the Luz group a long time ago. So we was doing more mechanistic study to finish that study. We haven't done that experiment. We did look into whether leptin have a function on B cell. We did a mixed bone marrow chimera when the leptin receptor was lacked from the B cell side or T cell side, which is more critical to have a vaccine response. We found in the T cell compartment is more important for the leptin signal. I totally agree with you. We cannot totally exclude the possibility the fasting or leptin supplementation can affecting other aspects of the immune system or even general metabolic system to affecting vaccine response. But because for most human study and mouse study demonstrated TFH is the major limiting factor for this. So we conclude the TFH function for leptin have a major role. I'm not saying this exclusive role for their irregular antibody response, but definitely that's a really good question. One thing I can say is for the other study, when we do the selenium supplementation for GPS4, we did look at the selenium supplementation in the GPS4 knockout mice. They don't have any enhancing function. So that's exactly what you propose can be tested.
Okay. My second question is related to the second part of the ferroptosis. So you mentioned that the reason you see the major contributor for the TFH death is because of the ferroptosis, is because the constant interaction between the T cell and B cell, right? So, but that doesn't explain why you do not see the effect of the B cell to transgenic mice. Meaning that, you know, yes, that the constant interaction between the T and B can contribute to the ferroptosis, but why there's no apoptosis? I mean, it should be as same as the other T cell death program. That's my question. So because you see that the BCL2 transgenic mice have no effect on the TFH.
Yeah, so you mean because they have a more germination B cells, they should also have an effects here?
No, I mean that because I mean that because there's major contribution or there's more interaction between T and B, which induce ferroptosis, that not necessarily means there's no apoptosis. But your experiment demonstrated clearly that BCL2 transgenic mice have no apoptosis. So my question is, why is that? Why, compared with other conventional T cells, why there's no apoptosis or very little apoptosis in the TFH?
Yeah, so that is a 50-50 camera. And so we use the 50 camera to look at the wild type and BCL2 transgenic in the same environment. And we didn't see the T cell from wild type or BCL2 transgenic have a major difference in the time point we look at. Maybe if we look at very long, they maybe have some level, but that is not the major regulator from the system we have been looking at.
So then I can ask if you can comment on what are your opinion on the role of apoptosis in the TFH? Meaning that, I mean, BCL2 maybe not contributed, which do you think there's like really little proportion of apoptosis participate in the TFH death?
We haven't tested extensively all the systems. They do have, I'm sure the cells, they can, based on different stress and conditions, they can be undergo cell death by different pathway. But in the physiological pathway we have been testing, we don't see the BCL2 transgenic is very strong protective for TFH cells. We see the GPF4 deficient is very strong, lead to the diminish of TFH. So from that experiment, what we can conclude, the ferroptosis is one of the major, and from this experiment, it's more important than apoptosis in regulate TFH survival and homeostasis. I wouldn't say if you culture TFH or in other conditions, they may have led to apoptosis, but in the physiological level, from what the system we have testing, we don't see apoptosis is the major contributor for TFH cell death.
So I think for me, the curious part is, or maybe it's something worth to think about, is your conclusion actually demonstrate two different pathways. One is you have dominant leading pathway to induce ferroptosis because of the constant interaction between T and B. On the other hand, you have second possibility. The reason the TFH is different, besides the increased ferroptosis, but also have decreased apoptosis. These things could be completely separate issue. I mean, you can't, I mean, it's probably not because you have increased ferroptosis equal decreased apoptosis. These two things could be completely controlled by two different machinery.
The apoptosis, for example, in T cells, I haven't studied this extensively. If you have a better idea and not more knowledge, I'm very happy to listen. It's like activation induced the cell death. Is that a main induced by apoptosis for T cells, for example?
Okay.
But yeah, so I think this is a context dependent. And for example, the thymus selection is really strong regulated by apoptosis, such as a beam knockout or BCL2 transgenic affecting the
sure. I think I'm actually specific talking about TFH cells, because TFH cells have this strong difference from the other T cell subset. Yeah,
whether there's a reduced susceptibility for apoptosis, I cannot say that. Yeah, and also, when we look at the BCL2 transgenic on T cells, they have a more memory compartment. So that means the effect cells, once they go into the effector phase, they can survive much longer as a memory compartment. So that means, of course, the memory generation is very dependent on regular apoptosis in T cells. But whether the Th1 or Th2 during their effector phase are very strong regulated by apoptosis, I think, at least at the moment, I'm not sure whether we are totally clear with that.
My last small question is, do you see the difference of ferroptosis on conventional TFH versus regulatory TFH?
Yeah. we didn't see much difference of the Treg cells or TFR cells regulate by GPS4 knockout in terms of cell number. I know there's a recent publication on Cell Report. They show Treg function is regulated by GPS4 deficiency. They demonstrate the GPS4 deficient Treg cells have more L17 production, which you will also see in a general study. Is like on knockout GPS4, we see the defects of TFA cells, but we see more GAT3 upregulation and more Rho-gamma and L17 production by T cells. Demonstrate there is a selectivity between the different T cell subset for ferroptosis.
Okay.
But yeah, that's a very good question. Thank you very much.
Thank you. Thank you.
Okay, good. I think Li Hao also has a question. Li Hao. Maybe I can just read Li Hao's question here in the chat. So very interesting talk. Thanks, Dr. Yu. Just curious about the fact that since leptin is regulated through circadian cycle, have you noticed that whether TFH or B-cell induced antibody immune response is also regulated depending on the circadian? For example, is the right time, is the night time more active? Okay, so that's the question.
Yeah, that's actually a very interesting question. Yes, the leptin for human being and mouse, they highest level during the night, and so that's why sleep is very important to maintain the leptin level. And I always consider if we have an overnight stay, whether I feel so hungry because our leptin level is not sufficient. So we do consider whether the circadian cycle regulate the leptin, and also leptin also have a difference between the male and the female. I have a student are testing this. So far, we haven't have a clear result, so I couldn't really answer yes or no for you. But if anything is relatively subtle, I haven't got any conclusive result, but this is something we are testing. Thank you.
Yeah, related to that, I kind of remember like there is someone. This is someone else, when you get vaccination and trying to get it done like in the morning rather than in the afternoon because of the immune response also have this circadian. But that might be something related to the antibody production and then the leptin-induced immune response. Well, actually, I have maybe a related question. So, have you considered like the therapeutic potential of leptin as a drug to like enhance the antibody production? Because I think in obesity, like leptin has been used in human, but not so successful. I wonder if in these clinical trials, like people have also monitored the antibody production or immune response.
Yes, thank you, Charlie. And that's a really natural question. There's a few thoughts here. The first of all, for the obesity, they actually have very high level of leptin. They have a 10-fold higher leptin levels than the healthy. Doesn't mean they have a better vaccine response. And actually opposite, some of the obesity from epidemiological study demonstrate they have reduced the vaccine response. So one possibility is like the insulin resistance, the obesity, and develop leptin resistance. This is well reported in the people studying metabolic system. So we haven't really started this. So, but I think it's less, as you said, the leptin treatment for obesity is also failed because they may have initial effects, but soon they induce the leptin resistance. So we probably don't think the leptin will be a good and directly, therapeutic approach to test in vaccine enhancement, but they can think about this other way to target the same pathway like STAT3 or mTOR pathway to enhance the TFH function for vaccine. I think that will potentially what we are considering. And also the other thing I want to make a point is, it's not to say that the higher the leptin, the better, even the leptin signal. And also physiologically, I think leptin is more covered the regulation. When they have a lower leptin, they restrain TFH activation. What we have discovered in the inflammatory condition, the TFH activation is more driven by like IL-6 or other pro-inflammatory cytokine instead of leptin. So I think it really depends on context.
Okay, that's great. Thanks. So next question came from Wen. So I wonder if when you want to speak up, or I can read the question. Okay, so maybe I can read the question. It is great talk. Thank you, Dr. Yu. My question is, what are the adjuvants used in flu and the HPV vaccine? Have you ever tested the leptin production in the different adjuvant treatments and monitor the TFH population?
I think you are basically very interesting with the adjuvant, and definitely adjuvant probably is so far the most important, and people always consider use a technical adjuvant for regulate vaccine response. For the HPV, the flu, I have to double check. It's a standard flu. I'm not sure whether they have, it's a inactivated flu. I'm not sure whether they need particular adjuvant. For the HPV, I think it's subunit HPV, but I can back to you. Cannot remember exactly the adjuvant used, but it's not the particular fancy adjuvant using for the standard vaccination. They do have a study demonstrate different vaccine and adjuvant can give a better and strong TFH activation. There's a paper, there's an experimental vaccine was published in Journal Experimental Medicine by Dr. Michelle Linterman last year. You can double check with that paper. I think there's a new adjuvant to demonstrate the TFH activation by this new type of adjuvant. The TFH activation is not just by leptin or cell death. It can also regulate by the antigen loading. It has been demonstrated the stronger antigen, prolonged antigen can enhance the longer TFH activation. And also it has been shown in the human, like IL-12 is potent to induce the TFH and IL-21. And IL-12 is induced by the antigen presenting cells, and this definitely can be regulated by adjuvant. But I'm not an expert with adjuvant. This is a very important question.
Okay, that's great. Okay, so this is very nice discussion. If there's no more question, let's thank Dr. Yu again for this wonderful talk.
Thank you, Charlie. Thank you, everyone, for coming to my talk, and would love to meet everyone in person at some stage.
Well, absolutely. I would hope like the border can be open. Okay, great.